Right, let's start the module 4.2, which will be talking about cell compartments in CompuCell. Okay, so remember that this meeting is being recorded and live streamed to YouTube. Okay, and the slide decks used for this module is already uploaded to the student materials, and we also be the first exercise will be done on based on a previous model. I'll give you the link for that model when you get there. Okay, so today we'll talk about cell compartments. Remember that you can use the chat and to ask questions. You can use the buttons yes, no, go over, go faster if you need to change the pace of the course. And raise your hand if you have questions, type in the chat, or just unmute yourself and just ask a question. We'll be happy to help. If you need a little bit more help, you have the breakout rooms where someone can work with you one-on-one. Okay, so today we'll talk about, we'll do two exercises if you have time, and we'll talk about the use of cell compartments in CompuCell and how to handle them, okay, and what does that involve, okay. So, as you all know, or you should know, that cells are not just a homogeneous blob of material. It has an internal structure, so it's spatially heterogeneous. So if you just look at a typical cell that you see on a cell biology textbook, you have a nucleus, a membrane, and lots of internal organelles inside. Okay, and the nucleus, it has usually a different stiffness than the cytoplasm. Is usually thought to be more stiff. The membrane have different properties than just the cytoplasm. The membrane is a two-dimensional fluid. You can think of it as a elastic sheet, or the cytoplasm is a fluid, but also a heterogeneous one. And when some cells do stuff, right, they're just not sitting there. So for example, when they move, they are crawling, they break their symmetry. There is a front, there is a back, okay, and the cells are very different, one side and versus the other side. Well, but even when the cells are sitting still and they form an epithelium, the cells are also spatially heterogeneous. The top of the cell is very different than the bottom of the cell. The apical side is in contact with some kind of luminal space with liquid and exchanging ions there. The lateral side of the cells are very sticky to the neighboring cells, so to make an impermeable membrane. And the basal side of the cell is touching the ECM, so it also has different properties. So in some situations, you actually need to model the cells that have they are not isotropic. They are actually have some kind of break of symmetry in themselves, so you can do other things in extend the scope of your simulation. Some process may only be reproducible in computer if the cells themselves are have this break of symmetry. Okay, so this is something that it can be done in CompuCell, and it has already been done to a certain extent. You can do compartments in CompuCell by basically two ways. You can also do it structural or functional compartments. And the idea is that your cell is not going to be made of one type of region, but multiple types of regions. These regions can be correspond to definite structures that you see in the cells. For example, on the top level, you have a cell sitting on a substrate, and I explicitly model the membrane of the cell in green, the cytoplasm of the cell in yellow, and the nucleus of the cell in blue. Okay, so you can model cells like this. The other way, you can do more of a functional or more abstract way to divide your cell, and what you see there on the bottom is the section of an epithelium. So what you're seeing is many different cells, okay, and each one of those cells have three different colors: a blue, a red, and a green. And in this case, the interpretation of this figure is that the blue should be the apical side that's facing the lumen. The base is the green is the basal side, which is seeing the ECM below, and the lateral compartment there is just to kind of separate those two, and you can increase the adhesion energies or the contact between lateral and lateral cells, so they actually are more sticky to the neighboring cell at that level. Okay. So, and this has been done and used in publications with people. The first person to use cells with multiple compartments was the group Andreas Deutsch, 2007. They used that to model a myxobacteria, which is a very long cell that moves, right? And they use lots of compartments, and they also link them together with springs, but that is for tomorrow. On the right there, you can also model lens formation. You have an epithelia, and not only have a stable epithelia, but you can change the parts of this epithelia at a given point and induce buckling. Okay, and in this case, by having the red compartments to pull another, and also the blue compartments to grow. Okay, and then you can induce this kind of formation. And you can also use these compartment models in a more even a more advanced way to model, for example, a three-dimensional epithelium. So in this case here is the tube of a renal of a kidney renal tube, where the cells have also a core region, a lateral region, basal region, and a apical region, and then it can also have the ability to secrete fluid towards the apical side and create cysts, as you see on the right. Those compartments, they do not need to be a kind of fixing space in the cells. They can be also very dynamic, and you can have compartments that can be anywhere. They can reorganize dynamically inside your model cell to actually adjust to the spatial cues that you have on the side. And that's something that I use in this toy model of the PCP signaling, or planar cell polarity signaling, on the Drosophila wing imaginal disc during development, where here on this right here you can barely tell the difference between the cells, but I just each one of those colors are different cell regions on the apical side with different kind of population of molecules, and they can self-sort out based on the neighbors. And because there's some kind of in this case they actually self-organize, and all end up being pointing out in the same direction. And this organization within the plane is known as planar cell polarity, and in the case for the Drosophila wing is used to actually orient the hair of that's spiking out of each one of those cells in the wing of the fly. Okay, so this I'll teach you how to do this kind of dynamic compartments in the last class, in the last module of this workshop tomorrow. Okay, so here you may have the kind of going on in your back of your mind that do you really need a module for that? Have you not done this already with Gilberto on the second day of the course, right? Because if you remember, you have done this exercise here, which is the of a crawling cell, right? So if I just let me try to play this again. If you remember, you start with an initial condition of 3 cells of different types, right? And you chose the energies such that they actually automatically sort them together. And what you did on this exercise is add some kind of external force at random directions in the lamellipodia, which is the green region, and that makes the cell to actually move in one certain direction. And I think the last thing that you were supposed to do in this exercise actually was to change the direction of this force, so the cell will move from one side to the other side and to the other side. Okay, so it seems that what I'm doing here is nothing new. However, there's a problem in this. There's an issue in this simulation that only arises when you have more than one cell, let's say. So here you have to get our nomenclature a little bit agree better on nomenclature. So far, I have been using the word cell to designate well a cell in our models, right? Right here, we are using three cells to simulate a, a biological cell, right? So when you get to these compartmentalized cells in CompuCell3D, what we usually do, we call these regions compartments, and the whole set of those three compartments as a big cell or a cluster. Okay, so when you are just having one cluster with three cell objects inside, there is absolutely no problem, right? You can just do that without thinking about there is no issues arising. But the problem here is, what now if you have two cells like that or four cells like that? Okay, if you just use the current plugins that you have been using so far, you have this following problem. At the beginning simulation, everything looks fine, right? The cells are behaving as they are supposed to behave, okay? But as soon as they see each other, you have a problem arising. Is that now those compartments, they do not know exactly to which cluster they belong, and they just start to mix together, interact according to whatever contact energies you set up at the beginning, okay? And if you have more than two, even more than two cells, the problem just gets worse, and things start to get ugly very soon. Okay, so when you want to use compartmental cells in CompuCell, you probably want to model more than one cell. Okay, and if you want to model more than one cell with different compartments, now you have to tell CompuCell, okay, this compartment belongs to which cell, this one or this one, or to which cluster each compartment belongs. Okay, so that is what you have to tell your code to make this distinction. Does this make sense? Okay, I hope it is. So I don't see the chat here. So if you have any questions or comment in the chat, please do it. I'll be keeping an eye on it. All right.
So they lose their belonging or their membership to the cell only when the cells get too close.
Yeah, once they start to get too close, they now they do not know how to distinguish anymore, right? So that's how I'll tell you how we solve this. Okay. So yeah, if you have a simulation where the cells are always far away from each other, then that's no issue. But there to be an exception and not the rule. Okay. So when you are using multiple cells or multiple clusters with different compartments, okay, then you need to distinguish between the compartments that belong to the same cluster and the compartments that belong to different cluster. Okay, and the way that you do this is by actually splitting the contact energy plugin into two. One will be the regular contact energy, which is going to be for cells of belong to different clusters. And when the cells belong to the same cluster, then we actually have to use a different set of contact energies, which and we do that with using the contact internal plugin. So here, the contact internal plugin that you can call from CompuCell specifies the energy between the compartments belonging to the same cluster. So, for example, nucleus and cytoplasm, nucleus and lamellipodia, and lamellipodia and cytoplasm. So, in this case, you have basically those three values here that you have set up. And usually when you have these models, you only have one compartment of each type inside the cell, so you usually don't use these diagonal values here. Okay, so this sets a contact energy between internal compartments. And in the case for the same model that you had before. For the, you can just use the same original values that you have for the original cell crawling model that you did on the second day. Okay, and now the original contact plugin um, set the contrast between compartments that belong to different cells. Okay, so now if you have two cells that come to contact, this is the values of the contact energy that you're going to set how this compartments here belong to this cluster interacts with the compartments belong to the cluster above. Okay, so well, in the case where we are actually doing a model, the same model as Gilberto did, but now for two different cells, the contact of the contact energies of those compartments with the medium can still be the same, right? The medium cannot be a compartment of a cluster, so that just stay the same. But those ones here, okay, they we have to reason out what those values should be. Okay, so does anyone has an idea of how big or small those values have to be compared to these ones here on the right? Okay, Ajay said much bigger. Sharon, bigger overall to avoid merging. Good. Okay. So usually we cannot avoid merging. Uh, any other guess? Do you should always do it bigger? Yeah. So as a rule, is those values should usually be bigger than those ones to exactly avoid merging. If you make it too big, the cells will never get close to one another. If you, it's okay for them to actually stay in contact with one another a little bit. You can make a little bit lower. If there are two kind of cell compartments that really want to stick to one another, you actually make those quite low. And if you have some kind of cell region that should really avoid this other cell region, then you can set that also very high. But the answer is that it should be bigger overall. In the case of the nucleus of one cell interact with the other parts of the neighboring cell, you want that to be very bigger because it should never, the nucleus of your cell should never interact with whatever structure of belong to another cell. And those values here also should be higher to avoid cell mixing, but probably not need to be as high as these ones. Okay. So, so with this little introduction, the objective of the first exercise here is to create simulation, just like the one that you did on second day, but using cell compartment features CompuCell3D. And the plan of action will be, first, you have to add the contact internal plugin to this code, specify the initial the initial cell, that all those initial cells that you put here belong to the same compartment. Okay. Then you have to implement cell cluster division. How you do this? Okay. You see that the cells are moving and then it divides. Okay, so I'll teach you how to do division of clusters instead of division of cells that you have seen so far. And then we talk about how to choose the appropriate contact and internal values, contact internal values of the cells. Okay. So we'll not write the code from the scratch. What I want you to do is actually to go to the link that I'm just giving you on the chat and download that basic code to either your desktop or to your NanoHub application. And I think you guys already learned how to do that. So I'll give you a minute to download the basic code, and we work on on top of it. Okay, so please let me know in the chat when you have that base code open and tweeted. Okay.
Julio, Hayden can email people the file too.
Oh, okay.
Yeah, I'll just send them to everybody. Sorry for blowing up your emails today.
Good question, Ajay, about that. Be the second exercise of this is how to choose those values if you want to create an epithelium. Okay, but yeah, you have to adjust those a little bit to get the correct values. The arbitrary, ultimately what matters is if there's some guidelines you can use, but in the end, you have to just simulate and see whether you got a well-behaved epithelium or not. Okay. So if anyone else needs the file email to them, please let Heidi know and he can do it. Okay. So if you have the simulation already, then I'll ask you to open this and tweet it and take a quick look at how what is being coded there, and just set to run and certify that it runs as expected. So let's see how many of you got into this. Okay. So if you have the code intuited and you run, what you should see is a cell with 3 compartments that wanders around in space. Okay. Hopefully, all the one that got the code in their compute desktop Nano Hub has exactly this kind of simulation running and no problem. Okay. Okay, Hayden will send a copy of the file to you. Okay, so I see that so I see that already 14, 16, 17 people has already done it, got it. So I'll just proceed, okay? And if I leave behind, I always leave the instructions of the previous steps on the slide, okay? So if you got the code, it should be running, no problem, okay? So what we need to do next is to add this content internal plugin, okay? So this is done on the XML level, and to do that you have to first in 3D select XML, okay, and then make sure your cursor is at line 60 of the XML file, because that's where you're gonna place the code. And you can just add new plugins to your existing code by going to the menu, selecting CompuCell3D, select plugins, and from this list of plugins you want to select the contact internal one, okay? Again, make sure that your cursor is at line 60, okay? You don't want to place a plugin inside another plugin, otherwise you have problems, okay? So once your cursor is inside line 60, menu, CompuCell3D plugins, and contact internal, okay? It has to be the contact internal. Don't use contact compartments, okay? That's old version. So if you click contact internal, then what you should see in the end is it just automatically pastes the new plugin for you and already specify all the pairwise cell type combinations for you, okay? And you should see something that looks like this in my screenshot, okay? So contact internal, and then all the values are 10 and the neighbor order is 1. Don't worry about them yet. Let's just leave them as it is, and we'll work on them later, okay? If you want to do something, you can already change the neighbor order to 4, okay? But once you paste the contact internal plugin, just leave it like that, okay? So if everyone has done with this, we can now proceed with our game plan, which is to specify what the initial compartments, that the initial compartments belong to the same cell, okay? You want to do that. So how you actually, what's the logic behind it, how that actually works inside the player? Is the following, okay? So I'm not sure if you make that explicit during the workshop, but every cell object inside CompuCell has a unique and unchangeable fixed ID, a number that it identifies them, okay? So it's like your social security code, right? Every cell has one that they say for them for their lives, okay? And this can be retrieved by at the level Python level by just typing cell.id, okay? If you have a hold on the cell object, commit cell.id, you retrieve this number, okay? So in a multicellular number, cells with multiple types, they can have same type, different types, okay, but they all have a unique cell ID. Okay, James just said posted an important message in the chat. If you had some issues because the file was not zipped, there is a zipped version of the file at the link that James just posted in the chat, okay? So continue here. In the case of the cell that moves, you can have those 3 compartments, and each one of those cell objects in these compartments, right, has a unique ID. In this case, it's going to be 24, 32, 107. The ID is a number, just like a social security number is a number, okay? So how do you identify cells that belong to the same cluster? Okay, that you do with the same thing. You actually add a second number that tells which cells belong to the same cluster or not. So the way to group those cells is by assigning a second ID called the cluster ID, okay? And initially all those cells have a unique cluster ID that actually is the same as the cell ID. And you can change that cluster ID to make a compartment belong to set which compartments belong to which cluster, okay? So all the cell objects with the same cluster ID belong to the same cluster. So in this case here in the bottom, you have, just by the image, you can see that there are two crawling cells next to one another, okay? Each one of those compartments cells has a unique number, 24, 32, 43, 59, 88, 85. But in addition to that, they also have this cluster ID number, which in the case for the cell outlined in red is, they all have the same cluster ID number 2. And in this case of this right cell, they all have the cluster ID number 10. So that defines that this group of cell objects form a cell cluster, and the second group of objects here forms another cell cluster, and they are distinct from one another. Okay? Does this make sense? If you guys have any questions or you need any help with getting the code, please let us know, okay? All right, so let's continue, okay? So with this brief explanation of how you actually distinguish cells belonging to different clusters in the code, we have time now to go into the code and tell all those three different cell objects that you have as the initial condition to belong to the same cluster, okay? So what you want, so this is done on line 10 to 20 in the Python steppable file, okay? And that's where we grab the cytoplasm and set its properties, its volume and lambda volume, the stiffness of the volume compression in the cell. And what you want to do is, after you grab the cytocell, so on line 14, okay, you want to add a new line to retrieve what's the cluster ID of this cell object. And once you have the cluster ID of the cyto cell, you want to now to assign this cluster ID to the lamellipodium and to the nucleus cell object, okay? So the way you do this is, again, you're gonna use the menu and the CompuCell3D Python list of functions. So what you do, you create a new line after the line 14. So while you are grabbing this cell here, you want to, right after this line, you want to create a new line and place the code right there. Okay, so after you create a new line after 14, you leave your cursor there, and then you go to menu, CompuCell3D Python, cell attributes, and you inquire about what is your cluster ID, okay? You do this, okay? You click, and you should place a code. There is some space you can remove, okay? The cluster ID of the cell, you go to the cell.cluster ID. Okay, so that's what you did. You ask what was the cluster ID of this cell, and then you store that in a temporary variable. Does that make sense? And now that you have this stored, you want to now reassign this number to the cluster ID of the lamellipodium and the nucleus. Okay. To do this, you have to actually create new lines after 18 and after 21. And on which one of those, you again go to CompuCell3D Python in the menu, cell attributes, cluster ID reassignment. Okay, so if you just look at what's written on those options, you can have a good idea what each one of them is doing, right? So you go here, cluster ID reassignment, you click once, and then you have to do this twice. And what it does is add those two lines here. Okay, you have to do this process twice. And the original code that it placed is just have a random number here. In my case, it's 1552, something like that. And instead of have this random number, you just set assign it to be the same number as the cluster ID of the cytoplasm. Okay, so you just set it to be. So what this command means is that you're going to reassign the cluster ID of this cell to be this new value. Okay, and this new value in our case is going to be this one here. Okay, so you do this for the lamellipodium and also for the nucleus cell object. And after you do that and you get out of this loop, all those three initial cells that you have at the beginning of the code will have the exact same cluster ID. Any questions? Is this making sense?
Does it follow the same process if you're initializing with multiple cells?
Yes, and that's what you'll be doing when you create an epithelium. In epithelium, you're going to create lots of cells at the same time, and you're going to go over groups of cells at different time at a step, and then doing the ID reassignment. Okay.
Okay, sweet.
Yeah. So in this kind of models, you have to do some work at the beginning of the code, right? At the start function to set up which cell compartment belong to each cluster. Yes. More questions? All right, so if you now save this code and just run it, you can just do that. Remember to save your code and play it and tell me if it looks right or not. And if not, why it does not look right. Okay, cluster ID not defined. So let me go back. So I'll leave the code. Okay, I know that this code here works. The cluster ID needs to have an underscore. Okay. So actually this variable can be whatever name, but share unaware from me, but the nucleus does not look entirely engulfed by cytoplasm. So it looks funny, right? Yeah, that's what I was kind of looking for. What's going on? Darshan had the answer, yes. We need to change the energy values, right? Because before the simulation, everything was doing fine because you're, well, it was the original simulation. And now that you tell that all cells now belong to the same cluster, it's not use the values that was in the contact plugin. It's now using the values that are in the contact internal plugins. Okay, so what we need to do is to probably Sharon, you got something that looks like this, right? Okay, great that you got it. Yeah, so yeah, it's kind of work, but it's not exactly what we're expecting, right? It's because we have to use the whatever values you're using here in the contact plugin. It's not using that anymore. Now the cell, because all the compartments belong to the same cluster, they are interacting via the contact internal plugins. Okay, so they're using these values here. And you see they are quite similar to the original ones by accident, but what you really need to do, you want to copy these ones here to the contact internal. Okay, so your next job now is to go to the XML and copy those codes, those values here to the contact internal. Okay. So what? And while you are there, you also have to change the neighbor order of the contact internal to be the same as the neighbor order of the contact plugin. They do not really need to be the same in your application. You may have reasons to use a higher one on one and a lower one on the other. Okay, but in this case, just because you want to reproduce exactly what you had before, you want then those two neighboring order to match. Okay, but you might have reasons why you have different values of the neighboring order for the contact internal and for the contact. Okay. So please do that and tell me if the cell looks better or not. Let me know when you are done with the this part of the exercise. I just sent the poll to all of you. Okay, for linear is looking good. Much better. In principle, it should be completely indistinguishable than the original model. Okay. So it should just look like that. But now what you want the cells to divide. Okay. So I'll just let people answer the quiz, how they're doing the exercise. And then we'll try to get into this part of the code. Okay. So I have here that at least 10 of you successfully completed the exercise. 2 are just watching. Okay. And realize that we have already kind of worked the contact internal values. So don't think that we need to finish with the cluster division and contact energies. Yes.
Hi, Julio. I have a question. So is there a way you can define different neighborhood order for your different compartments? Because I can imagine the nucleus doesn't really need a very big neighborhood order because it needs to stay within the cell. So if you can do that, I guess that makes some sense. Does it make some sense? Like having a lower neighborhood order for your nucleus
For
compared
different
to...
objects. Yes, it makes sense, but that's not we don't have that flexibility right now in CompuCell3D.
Okay.
Maybe something that you can do on the C++ level, but as far as I know, the only thing that you can really do is have a different neighbor order for the contact and a different one for the contact internal.
Right. That's what I
Yeah.
was thinking.
Yeah,
Cool.
but good question.
Thank you.
Yes. Just a general question: why are these addition values so sensitive? Because they control who the cells want to be in touch with, right? It's the way that it behaves in the model. And we're using values of 10, 20, right? And the temperature is 10. So and of course, when you do the sum, you're summing usually more than one. So they end up have a very high end value in the energy function. Okay, so I think that a lot of people have already finished the exercise. So what I'll teach you now is how to implement cell division. Okay, so let's go back to Tweeted. Okay, and you want to usually as a good practice, you want a different steppable for every kind of different feature or behavior that you want to add to your code. Uh, what many people do at beginners in CompuCell, they write all their functions in the same steppable, and that's just not good coding practice. So here we have already a steppable, a built-in steppable that just sets the uh, initial configuration of the cells and make the cells move around the directions. Okay, now you want to add a functionality that you're going to make the cells divide, and we want to add that as a new steppable instead of reusing the old ones. Okay, so to add a new steppable in CompuCell, the tweeted editor can do that for you. You just select the Python steppable file here, right click on it, and select the option add steppable. Okay, it's that simple. So go back to it, select the steppable file. You select the other Python file, not going to work. You have to use the last one that has the name steppables at the end. Click add steppable, and then you have a new window. You have to give this steppable a name. You can give whatever name you want as long as it makes some sense. Okay, in my case, I just said the steppable name is going to be divide cluster cell. Okay, and what's really important here is that you can have different types of steppables. Okay, and usually we're using the generic one. When you want to do cell division, you just use mitosis. Okay, but when you want to divide a cluster which is made of multiple compartments, okay, you do not use mitosis. What you use is the cluster mitosis steppable type, because CompuCell has some special functions that look at how your compartments are distributed inside the cell, and when it divides, it reproduces the spatial distribution of the compartments into the daughter cells, which is very cool. So please select cluster mitosis, give whatever name you wish. You can leave the call frequency to 1. You can give a different call frequency, but that's all you need to do. Okay, so again, names, all variables and things in the code cannot have to follow these rules here. Okay, so you do this, and what the 3D does for you is just create the new steppable for you with the name that you gave, and is a mitosis steppable cluster based. Okay, so if you go to your code, which should be around line 50, okay, you should see this new code pasted for you. Did all of you got it? Did you get something that looks like this? Okay. Yes. Okay. Thank you for answering the chat. So what's going on here? Okay. So you see that there are a lot of things happening in the step function. Okay. This has basically 3 parts. This code. This very first one here is not really needed to do anything. It's just showing how you can handle cluster at the Python level. Okay. So here you have a cell loop over cells, and given one cell object, you can actually get the list of all the cluster, all the other compartments that if the cell belongs to a cluster, you can retrieve a list of all the other compartments that belong to the same cluster. So this is what line 56 is doing. You use this function and add the cluster ID, and you're gonna list of all the other compartments that belong to the same cluster of that cell. And then you can actually loop over all those compartments, right? And see who are these compartments. Okay, so this is just placed here to show you how you can handle clusters at the Python level. Okay, so from a cell you can get the clusters, and you can also get a list of clusters, and then you can loop over all the cells that are inside this list of clusters. Okay, the second region here, from here to here, is the code used to decide which cells you divide. Okay, and the last group of code here is where the division actually takes place. And if you just take a look at it and remember how we did this for the mitosis exercise, is exactly like before. It's just that instead of having a divide cell random orientation, you have divide cluster random orientation, and you can also have these other options. Okay, specific axis along the major axis, along the shorter axis. Okay, so okay, this is just what I said. Okay, there's a question in the chat. Why does the new steppable come with a start function? Never needed. Usually don't need it, because it assumes that whatever setup that you have at the beginning has been done on a separate steppable. But there's nothing that prevents you from adding a start function in your new steppable. Okay, so if for some reason you need that, for example, you want to add some kind of tracker to how many divisions happen in your simulation, right? You probably want to set up at the start function a dummy variable that you're going to just be used to count how many divisions you have. Okay, so you can write the function right there for you. Easy. You have a question?
Yeah, we in this function we're cloning the attributes to the child cell. Do we not need
Yes.
to reassign a new cluster ID?
No, it does automatically for you. That's the
Oh,
beauty
awesome.
of it. Yes.
Perfect.
Okay, more questions. So I already explained this, but very quickly, this loop here is going over all the cells in your simulation, this, and then you, for example, if the first cell that you got was the cytoplasm, this here, you give the cytoplasm cluster ID into this function, and you get the list of that includes all those 3 compartments. And then what you can do, you can loop over this list to get the cell, the cytoplasm itself, the lamellipodia, the lamellipodia, and cytoplasm cell. Okay, so and then because you're looping over the cell, then it does from this one, and then you get all those 3. So it's just showing how you can from a cell go to the cluster and go to a neighboring compartment, and then from this neighboring compartment go back retrieve all the other cells. Okay. So here on the next set of block of code, which is actually do the division, we do not want to do a division based on growth. Okay, we don't want to. We want the cells proliferate, but you do not want to waste time making all those cells grow in accordance with the respective relative volumes. Okay, you want to do something that's way more simple, which is just to tell the cell to divide a specific time. Okay, so instead of having a growth-based division, we just want to make a division at a random event. Okay, so what I want you guys to do here is on this line where you have if cluster volume is bigger than this value, you just replace that by just calling a random number and check if it's below some kind of value. So I suggest using check if this random number is below 0005. Okay, so please replace this condition with this one. Okay, so I can tell you what's going on here. You are going over, you get the list of all the clusters in your simulation, which in this case is going to be just one. Okay, and then you're going over all the compartments in this. So you have the list of all these clusters. Okay, and then you go over the cells over this compartment list, and then you are here. You're just adding up all the volumes, and also you're retrieving the cell ID, cluster ID, and if the total volume of the cluster is bigger than some value, you divide. Okay, so that's what the original code was doing, but what I want to do is to just change this condition to be a random event. Did everybody got it? Got this? Yes. Okay, good. Now I have already told you the next set of lines. You actually are dividing your cluster. Okay, so once if satisfy the condition, okay, you add that cluster ID to. Sorry, I should have to go back a little bit. So right here at the beginning, I create an empty list with is the list of clusters that should divide at that time step. Okay. And then if satisfied the condition, you add that cluster ID to that list. And what you're doing on line 76 here in my code, okay, is you are telling the CompuCell to divide that cluster. So you notice that you're using the command divide cluster at the random rotation, and then you just give the cluster ID. But you, as before, you can also give different options to how you decide the division plane. Okay, so as I said, what the code already does is, in this case, just choose a random direction of the cell that you should divide. So for example, it chose here, and what it does, it divides the cell and copies the compartment distribution to the two offsprings, and what you have in the end is now two cells that has the same number of compartments within the same spatial distribution with all the same properties of the cells. So all the cell properties are cloned from the mother cell to the daughter cell. Okay. So one thing that our code already does for you is that the code is assuming that this is a growth-based division, right? So what it does in the end is go over all the compartments in that cluster and divide its target volume by half. Okay, because we are not really growing the cell, we have to turn that off. So those two lines here that go over the compartments and divide the target volume, you have to ignore, or tell the code to ignore, and then you can either delete those two lines or just comment them. Okay, so please do that, otherwise as your cells divide, you're going to get smaller and smaller and smaller until they disappear. Okay, we don't want to do that. So what's going here on the update attributes, actually don't even need this line here. It's just first fixing whatever parameters of target volumes. If you have a target surface, maybe also need to adjust that. And then on this line here, you're just cloning one cluster to the other, the two offspring clusters. Okay. Okay. Is this making sense so far? If it is, please save and play the simulation and tell me if it looks right or not. Okay. So I'll go back to the previous slide and let me know if you how is it going. Okay, so right, have an answer that the cells do not look right. Isn't that funny? That sometimes happens, the lumbar reporter run off by itself. It happens with me too. That's after division, right? Yep, okay. So at least have two testimonies that it's not looking right. Okay. So let me just relaunch the put, see how you guys are doing. Same with me, okay. You're not getting any division. Well, it's a random event, so you maybe you set the random event to be true. A very low probability, you can just increase that number and redo the simulation. Sometimes, well, because it's random, sometimes with the same simulation you get division right away. Sometimes you get a very long time for the first division. Is it better to choose division along long axis? I'm not sure, because sometimes those cells are very elongated, and the one time I tried to do this, sometimes the division, the reproduction algorithm is not always perfect. Sometimes you can miss a cell element. Okay. Ideally, what the cells do, they stop crawling, and then they round up, and then they divide, right? Here we are just doing the division right away in a very unrealistic manner. Okay. But usually, even when the cells are in epithelia or even mesenchymal cells that are elongated, when they divide, they round up, but they still divide along the minor axis, and they have some that called the Hertig's rule. They do kind of have a memory of what the shape they had before division. Okay, so that happens. The cells round up and divide, but after they round up, but they do not lost the memory of the shape that they had before, which is very interesting. Okay, there are some molecular fingerprints that stay in the cell for a longer time, and that guides its division. Okay, so it seems that most of you are, at least one person needs more time. Okay, but I don't know when that was clicked. But most of you are done with the exercise, so you probably got something that looks like this, right? The cell divided, yeah, the division is working, but I'm not sure anymore if it's working or not, because the cells start to look very funny. Okay, why it's looking funny like this? Okay, I already asked 3 times why it's not two times why it's not looking right, and the answer is actually the same as the previous 2 times. Self-related surface energies. Yes. What exactly? What exactly forgot to do? Okay, I'll give the answer. We chose the appropriate internal contact values, but we did not reset the external contact energies, right? We just leave at what you had before. Okay. So what you have to do is you are still use the same values that you had at the beginning when you start with the single cell compartment simulation, right? So what you have to do is go back to the contact plugin and rewrite those 6 contact energies according to what we all said in the beginning, that they should be slightly higher or much higher than the internal contacts between compartments. Okay, does that make sense? So that will be the last task of this exercise. Go back to the XML code and choose an appropriate set of energies until your code looks fine. Is not affect the internal structure of the cluster. When you have just one cell, it looks fine. As soon as you have cell division, now you have 2 cells next to one another, and those compartments are interacting with one another via the external contact energy. And because they are still set to be as it was an internal contact, they are glued together. Did it make sense, my answer? Okay, Ajay and Ivan make sense? Okay. As soon as the cell divides, okay, now they are interacting with the external contact energies, and that has not been set up. Yeah, maybe something like this. Okay, so we have to reverse the magnet there to actually can repel. So I already tell you, thing those three values can be the same value, and those three can be the same number. It should work, okay. And let's see how I'm going of time. I have, well, we still have 50 minutes according to the original schedule, so I think we're doing okay. So did anyone got a very nice looking simulation? If you did, please share with us in the chat which values of energies you used. Okay. So this can be the same number, and this 3 can be the same number. Okay. Tell me, tell us, what did you choose? You can even use the Slack and add a snapshot of your simulation. That'd be fine in the Slack channel. Okay, James said 1,000 for all the nucleus, and the other one said 500. Okay. So James said it shows the values are not of magnitude higher than Barrett. So let me see if there's anyone posting pictures on the Slack channel. No. Okay. So okay. So of course they're not absolute right or wrong in this case. As long as the cells do not merge one another as they did before, you should your values are reasonable. Okay. The ones that I choose was 100 for the nucleus and outside interactions, and 50 for the other cell compartment interactions. Okay. So Liliana, sometimes the division does not work perfectly, and you can get cells without nucleus, right? So that happened in my simulation too, which probably tells us that we have to either add some kind of error correction mechanism, or we should tell the cells to stop moving, round up, and then do the division in a more not rush up manner. Okay. But yeah, it happened. I think in this simulation that I'm showing here on the screen, that happens at the very end with a cell that was around here. I'm not sure anymore. No, I stopped the movie before that, but it happened to me too. So if I use these values, you should see something that looks like this. Okay, how much division the cells have before they well, here is well, if you choose the division event to be very low, then it will be moving a lot before it divides, but you should see something that's kind of similar to this. Okay, so this ends up our first exercise. Okay. Okay. So this I'll send me a poll for this first exercise. Okay. Please answer. Barrett, is met remove those clusters that are missing a component. So yeah, that could be a way to correct it. But you guys ask very good questions and good ideas. Yes, that would be a way to do it. But the objective of this exercise is to get for you to see what it takes to make multiple cells with compartments. Okay, you have to differentiate between the contact and external contact energies, and I try to do in a way that you got a lot of wrong results along the way to show where the typical kind of mistakes that we do as we try to do this process. Okay. So is there any question about this before we proceed to the next one?
I had a question.
Yes.
What if you wanted to have a non-compartmentalized cell transform to like a mesenchymal cell that was not compartmentalized to transform under some condition to a compartmentalized cell, or for one cell to divide to give one compartmentalized cell and one non-compartmentalized cell?
Okay, very good question. So one thing that you can do is you can tell the cell to divide, and then in the update attributes of that cell division, you tell the two other cells to actually share the same cluster ID. And then after division, the cell will now be a compartmentalized cell.
Right. Okay.
And of course, if you want more than that, you can divide 3 times in a row, and then you can have then 4 compartments in your new cell. So that would be one trick that you can use to convert a single cell into a compartmentalized cell from one step to the next. Does it make sense?
Yeah, so there's a simulation that we are building where there is a bunch of cells that are represented just as single things, that they're like mesenchymal cells, and then there's a wave that is slowly passing, so that as the wave passes, that cell that was mesenchymal has to turn into like an epithelial cell, which I normally represent with apical, lateral, and basal compartments. So I have to reassign it to a very particular type of cell.
Yeah, you can tell that cell to divide 2 times in a row, and then every time you divide, you
Just assign.
reassign the cluster ID. So you have to do
I
some code
see.
on the Python level, but yeah, that's one way to do. Or instead of using the cluster ID, you just on the Python level you can kind of divide your cell into compartments. There are multiple ways to do. The division will be the most straightforward way to do it.
Okay.
But it can be done.
Yeah.
Can you also play with the target? Yeah, you can, of course. Then now that you have the code, you can do whatever you want as a homework, right? Is there a way to take 2 separate clusters merging? Yes. Maybe you can make a condition that if the 2 cells of different clusters see each other in a particular condition, you just go over all the compartments of one and all the compartments of the other and make sure they have the same cluster ID. That would be a way to merge. Yeah. Easy.
Actually, I don't know. Would you be able to divide between one cell type with, say, 2 compartments into a different cell type with different compartments?
Yes, there's nothing that prevents you to do that. It's just a question that you're gonna take you some coding to do that, right? So the beautiful thing about have this Python level on your code is that you can do all this kind of customized stuff. You only have to be careful about how you code it. Very good questions.
Okay, thank you.
So let me try to quickly go to the next one, okay? So what I want you guys to do is to let's make a code a simulation of epithelial cell, okay? So using a functional kind of use the cell compartments and clusters to make a functional difference between cell regions, right? A more abstract way. So you can do that to do this compartmentalized cells. So here red is apical, blue is basal, and green is lateral. And then once you have this, you can actually use this code to actually create some kind of furrow formation as you see here, okay? So this simulation here actually uses spring force between cells that we're teaching tomorrow, but the objective today is to just create this basic layer of epithelial cells above a ECM-like object below. Okay, so let's proceed to this second exercise. So please let's go to 3D and create a new CompuCell3D project. Okay, you guys should already know how to do this very easily. Let's call it module 4.2 tissue folding. Okay, the tissue not fold today, but tomorrow it will. Okay, Python plus XML. Here, this is important. In this new simulation window, I want to make X 350 and Y 100. Okay, and the X boundary condition should be periodic. Okay, so I want to go over on the X and non-periodic, no flux on the Y and Z. Okay, so this, and here rectangular slab. Okay, then because you want to do an epithelium with an ECM on the bottom, we need all those 4 cell types: apical, lateral, basal, and ECM. Okay, you have to type here and push up. Okay, once they are all here, you hit continue. Make sure that you have all those 4 here, because if you miss one, it's very hard to fix that later on the code. It's cumbersome. Okay, so ignore the chemical fields, and here on the last window of the simulation wizard, let's select contact internal, volume flex, center of mass, and cell neighbors. So please select those 2 here, this one here in the middle, and those 2 on in the last column. Okay, select those 5 options here. Do this, hit continue, the configuration is done. Oh, sorry, ignore what's written here. You don't need focal point plasticity. Don't use that. Okay, so up to here was right. Contact internal, volume flex, center of mass, and the cell neighbors. Okay, so this is just telling you that you will be tracking who is neighbor of whom. Okay, so if this is done, hit continue, and you should. And now the challenge is to create an niche condition that looks like this. What you want is a row of cells of type apical, then a row of cells of type lateral, and then a row of cells of type basal, and then below a row of cells of type ECM. Okay, so in your code, at the very end, you have the uniform initializer, and you have that snippet code of where you define the uniform initialize the region of box minimum, box maximum. What you want to do is want to have the X going from 0 to 350, which is the X size of your lattice of a region, and the Y to be about 1 cell width, which is going to be, let's make the cells 7 by 7. Okay, and you do this only 1 layer for the Y, so you have going across as just 1 cell layer, and then you repeat this code, this region, so start region and the region here 4 times to create those 3 layers of epithelial cells, and then the bottom layer of ECM cells. Okay, if you want to do the ECM cells, can be 7 by 7 as well, but if you want, you can also do them as 35 by 35. Then you have 10 big ECM cells. Okay. So doing this might require a little bit of time. I'll give you guys 2 minutes to do it, and then send the poll to see if you were able to create this initial condition. Okay. So I'll launch a poll here to see how you're doing with the exercise. And I already paste here on the screen my solution on how you do this. Okay. I basically on the informationalizer I make 4 calls to create each row at a time, and I made the same row to be very big cells of size of width 35 by 35. So you can just copy what I have here on the screen, and that should give the initial condition. Okay. So that goes to answer one of the questions that I have in the chat. What if you want to start with multiple cells? Okay, that's a little bit tricky because you have some criteria to identify which cells belong to the same cluster, right? And I guess you can see what the criteria we might be using for this one. Yes, you can keep ECM as one cell. Okay, since that a good fraction of you have already done the exercise, if you use 35 by 35 cells like I did here, you have to make sure that the target volume of those ECM cells is 1225. Let me double check here. Otherwise the cell will shrink as soon as you begin. Okay. But yeah, if you do a lot of cells, 7 by 7 cells, Wait. Your question was not if you can use ECM as small cells. The question was, can I keep ECM as one big cell, 350 by 35? No, that's when you specify the width, right? This value here in the box initializer, the width is the same for the x and the y directions. Okay, you cannot specify a length and a height and a depth separately. So if you try to do one cell, you actually cannot. The best thing you can do is to try to do 35 by 35 cells and have as I have on the screen. Okay. So I will proceed. So this actually is the part that takes the longest to do this. And so if you just set up and run your simulation, if you forget to, if you use big ECM cells and forget to adjust your target volume to be also big, they will shrink, and what you should see is the whole epithelium kind of falling down, right? But the other thing that you have to do is want to group the cells to be together to be of the same cluster, right? So now if you just run simulation, as you fix that, it will look like this. It's gonna be just, you treat those different cells as separate cells. What you want to do now is group them together. And how you can do that? You can use the fact that initial condition the cells are right on top of one another, and you can go over all the cells of different types and check if the center of mass coincide. And if the center of mass coincide, you can assign them the same cluster ID. Okay. So that's here. You do loop over one cell type, and then you do a loop over the other cell type. Check if the x coordinate of the center of mass are equal, and if they add, you use the code to reassign the cluster ID. Okay. So that this is one way to group the cells together. I just, because you're already over time by 8 minutes, I'm just placing the code here for you. So this has to be done on the start function. For example, I start with the cell on the top, the apical side. I do a loop over them, and then for each one of those cells, I do a second loop. Okay, so I do the visit, the loop again, but notice that the dummy variable has to be different than the first one. You cannot use cell here and cell here. So I call this the second cell, and then I do over all the lateral cell types, which could be the green one. Okay, and then what I do, I check if their x center of mass coordinate, so COM stands for center of mass. I check if they're equal, and if they're equal, that means they are on top of each other, right? And if they're on top of each other, I can assign this second cell to be the same cluster ID as the first cell. So see that if I'm setting the reassign the cluster ID of the second cell here, and I'm putting there the cluster ID of the first cell in the loop. And of course, once I grab one of those red cells, run over the green cells, and as soon as I find the one that's below it, I reassign, and there's no need for me to keep going over the loop because I already found the cell. So actually I use this comment here break that tells me to, okay, once I found the cell that's below me, I don't need to look for the cells anymore. I can break out of this internal for loop. And then what you do that for the lateral cells, and then you can also do that to match the apical side with the basal cell that's below. So you just repeat the code likewise, what you do for the basal type of cells, and you reassign the cluster ID. Okay, so I forgot to update these figures here in the bottom, so please, you may have noticed that you have a white line here. Just ignore that for now, okay? In your code, you will not have that white line. But if you code this, you should have grouped those cells together. Okay, so let me know how you guys are doing with this part of the exercise. Okay, okay, Darshan had a problem with in NanoHub.
So Darshan, the
Yes.
easiest fix we have for that is if you if you terminate all of your active sessions, log out of NanoHub, close your browser, and then reopen and log back in. It should help stabilize it at least for a little while.
I'm sure I'll try that.
Let me know if that doesn't work.
Is it clear what's going on this part of the code? You're just matching the X center of masses as I criteria to group cells together with the same cluster ID. Any other general question about compartments that I can answer in the meantime? Please feel free to just unmute yourself or write something on the chat. Okay, I can share in the end the code that I used to do this. I just want to try to finish up this exercise in the next minutes. Okay, so what you're doing here, we set the initial condition. Okay, now we have to assign the group cells together. Okay, all this is kind of basic CompuCell code that is not necessarily teaching you much, okay? But once you group the cells together, and then it's time for us to actually choose the contact energies that those internal compartments should have and the external compartment interaction should have, right? If you want to make sure that your cells are actually grouped together, one thing that you can do, you can on the player, and if you click on configuration, and you can set up the cell boundary colors. Okay, so you can have a color for the cell boundaries and another color for the cluster boundaries, and you can turn on and off the rendering of the cell boundary versus the rendering of the cluster boundary. Okay, so what I like to do is when I have simulations with clusters, I like to turn off the cell boundaries and have only the cell cluster boundaries turned on. So then what I have, I get rid of the white line between compartments, and I only have a line around the clusters. Okay, so that's one thing that you can do in the player to visualize if you group the cells to in a correct manner or not. Okay. So once you get this to work, then it's time for us to choose what should be our contact energies and internal contact energies. Okay. Uh, so does anyone want to tell me what the relative values, if which of those values should be low, which should be high? So if you have not done the exercise, let's suggest you to stop and let's just kind of think about what the contact and contact internal energy should be, okay? Because that is the modeling part of the exercise where you have to make a decision as a modeler. Okay, Ajay said that apical and medium should be low. Okay, why? Because it should be facing, you like to face that side, right? Okay, what else should be low by the same criteria?
The basal to the ECM.
Yes, very good, because you want that to be, right? So
Yeah.
these are two things on the external contact energies. What should be very high on those two, on the external contact energies?
The basal to the medium and the apical to the ECM.
Yes, they should be very high because you don't want the cell to be flipped, right?
Yeah. Yeah.
So yeah, the way that you proceed to add those parameters is just by reasoning out one at a time. Okay, what should be high, what should be low. So my suggestion is always to do not write numbers right away. First, start write what should be high, what should be low. Do that in a piece of paper, okay? And because you do not know how many levels of high and low you should have yet. So in the internal contact energies, what should be very high and what should be low? For now, for this table here on the right.
Blue, green, low. Green, blue, low. Red, blue, high.
Yeah, red, blue, right. The apical and the basal compartments should not like to touch one another, right? Because they should be on opposite sides of the cell. So that will ensure you that the basal side will be opposite to the apical side, right? So that's very good reasoning. Okay, so this is one way that I reasoned this out. We was not necessarily on this order, but that's okay. So I said that the internal compartments it seems to be cohesive, so we want them to actually be low, right, between them. What else? There are no cells with medium or ECM, right? So we can ignore those values here. You don't have a cell that has an ECM compartment, so and you have no cluster with a medium compartment, so these 10 values here can be ignored, okay? And at the same time, we don't have a cell with two apical compartments, two lateral compartments, two base compartments. So you also can ignore this diagonal here, okay? Is a pretty common stability with adding additional energy terms that some cluster components, usually the nucleus, disappear. That really depends on how you chose those additional energies. I'm assuming that your question is when you're just setting up the cell without cell division. If you have a compartment that is very small and you make its additional contact energies to be too high, you can accidentally induce a disappearance of a compartment. Yes.
Don't you want B to A high?
Let me see. A and B high. Yeah, that can be higher.
A to B high.
Yes, yes. This one here, right?
Yeah.
Yeah, we just reasoned that out, and Sharon also said reason this out. Yeah, that can be higher. So I probably should. Yeah, that was. Uh, yeah, I should update the slide. That this should be high. Okay. Yeah, I agree with both of you. Yes. So this actually sets up all the contact internal energies. And now let's look at the other ones. So the apical kind of should be a little bit low, plus, right? Then these other ones, I said the cross-sectional energy, so like lateral to this one and lateral to this one should be relatively high. This one to this one should be very high, right? Well, as high as this one at least. And likewise, you want your cells apical align on apical, lateral, and basal. So you want this to be also relatively low. But you definitely want this to this and this to this, apical to medium, and basal to ECM to be even lower. Okay. And the opposite case to be very high. Okay, so I forgot the way I went over the slides, but here I took the apical side and said, okay, you want the apical to medium to be low. That was the first values that we agreed in the beginning, and the apical to the ECM, right, to be very high, to prevent the cells to be flipped. And then likewise, the same thing, the same reasoning can be choose for the basal, medium, and basal ECM. And then I just fill up the other ones. Okay. ECM can be 0 because they should be the same cell or the same object. And medium with ECM should be very high. They should not never touch one another. So what I'm doing here is I'm just kind of looking at the tables, looking, okay, what should be my configuration? What should be the energies that should give me a stable configuration, right? Reason that out and said what should be higher, what should be lower. Okay. So what I did here, if you can go over my slides that is on the drive and see how I reason out all these energies. It may not be perfect, it may still need adjustments, but what you have to do when you're playing with external contact energies and internal contact energies, you have to do this kind of reasoning out at the beginning, and that should give a good starting point. And what you definitely do not want to do is to just place numbers there and then start guessing those numbers blindly. Okay. Okay. So here is a set of values that I use, and that gives a reasonable stable epithelium. Okay, so you can try your own numbers, but I want to just share this, and you can try by yourselves, and you can download this number again, those slides, and copy. I just want to finish with this exercise. That's actually everything that I have planned for today. And it would be good that you actually do this exercise to the end and have the place code, because we actually want to use this code tomorrow to actually add some kind of spring forces between compartments and do some tissue folding. And I know that some of you want to do that for the hackathon. Okay. So with this, I end up this module. If you have more questions, please send to me. And I think Hayden has some announcements to make before you disband, so please wait for him before you turn off Zoom. Hand over to Hayden. And thank you everyone for your patience and sticking with me till now.
So Julio, let me say something, because there seems to be a problem that many people are having, which is that you generated your code with the newer version of CompuCell, And CompuCell checks the version number and throws an error if you run it on an older version. So you could hand edit to change the version number in the XML back to an earlier version. Or people who are using it on NanoHub, if you go into the XML when you've loaded it and change the version number by hand in the XML, perhaps Julio, you could show them where that is, then you won't get an error. And it should be a warning rather than an error. It should run, but if it's not, then you simply need to go into the XML and change the version number. Okay. So you see where Julio is pointing? If you change that number back to an earlier version number, then you will not have a problem. Is that clear for people? So if you type 4.2.1, then it should work. Please try that. And Hayden, maybe we should send out an email about that.
About what? Sorry, I was checking something else.
Okay.
You can fill me in.
Okay, I will. That's a good question. Some people seem to be confusing warnings with errors. So if it's a warning, it'll say okay, and you'll have a button and it will execute anyway. If it's an error, it will not allow you to go forward. So it'd be helpful if you'd tell us, is it an error or a warning that you're getting?
I'll share other thing, James. Sometimes there are two places that actually specifies the version. One of them is on the XML, and another of them is on the .cc3d code file here. If you click there and open with Notepad, that also says the simulation version. Okay, so if you want to get rid of the warning, you have to change that both in the XML and also right here.
So that means that our instructors need to be careful to build the code on NanoHub so that you don't, or hand edit it so that you don't have an incompatible version. So you have to roll back to 4.2.1.
Okay.
If you can find a fix for that, that would be good. There's a question. Is he?
Yep.
Go ahead.
I was just wondering. Sorry, is now a good time?
Sure.
Cool. So for compartments, in terms of good practice, if you are wanting to go for each cell and look at the different compartments, sorry, each cluster, and look which is like representing one cell, and loop through the different compartments inside it, are you better to go like start looping through cells and then find the cluster ID and then find like match that with the other cells with that same cluster ID, or loop through clusters? If that makes sense. because I found the 4 compartments in self.clusters.
Yeah, that's a good question. I'm not sure what's the best way to do it. You have maybe want to try both and see what looks less,
You can. You
what looks
can.
cleaner in the code. I'll let Pedro answer.
If I'm not mistaken, there is a function where you get the cluster and then you access the compartments that belong to the cluster. You can loop over them individually.
Okay,
I
sweet.
think
That'll
I
be
showed
the
up in the code here. Oops. Here, if I'm not mistaken, sorry. So if you the call self cluster list, you go over the clusters, and then you go over the compartments inside this
yeah.
clusters. Okay.
Yes, exactly.
Yeah, I
So
think it should
just
have
to
been called cluster
clarify,
list clusters instead of compartment list.
yeah, just to clarify, so your cluster list is got that is all your cluster IDs, and then when you're looping through, yeah, because you've got each cluster ID will have like one cell, which is like made up of 3 compartments. So does it loop through each cell or each individual compartment of all cells?
Can you repeat the question?
So you're looping through your clusters. So say you've
Here.
got
Yes.
five clusters.
Mhm.
Each one of those clusters is made up of 3 compartments. So, oh no, I think that kind of makes sense. Yeah, you're looping through your clusters and then you're looping through the compartments within each cluster.
Yeah, that's what it's doing here.
Yeah.
Here and here.
Yep. Okay. Sweet. Got it.
Yes. Okay, good.
Thank you.
No problem.
Julio, if you don't mind, if I could just temporarily break in here for the announcements, because we've
Sure.
told people to stay, but it's late and I won't take long. So I don't want people just to be waiting for a 2-minute announcement, and then we can get back to Q&A once we're done there. Let me take over the screen share for just a second, if you don't mind. Okay, so I just wanted to quickly go over the schedule for tomorrow and a note about parallel sessions, since we obviously had a little bit of a technical hiccup today. So tomorrow will be our last day. This is actually not to be determined anymore. This is from an old slide. So we will start talking about links first thing tomorrow, like Julio said. It's an extension of a little bit of an extension of what we've been talking about here. And then Priam will give us some examples of links in epithelial cells, as well as talking about tissue folding phenomenon. Again, we'll have about 45 minutes of open hackathon discussion time now that teaming has kind of gotten underway, and preparations for tomorrow. We'll have another round of parallel sessions. This time there will be 4 options, and we have to generate new links for these, obviously, since the links today didn't work. I just realized I don't have my video turned on. So we will, in the morning announcements tomorrow, we will have those links ready for you. The 2 sessions, so session 1 and session 2 are both external guest presentations. We have Shane Hudson from Vanderbilt looking at a computational model of secondary palette fusion and disruption. This is a very interesting model, and the code is huge. So it's interesting from the perspective of it being a very complex model that was written in older versions of CompuCell and has been updated to run in the new version. So you get an up close look at kind of a gigantic CompuCell file tomorrow if you go to that session. Then we have Mrunmoy Mukherjee who was looking at cluster size, distribution of cells disseminating from a primary tumor. So if you have a hackathon project that's interested in cancer modeling, this one would be a good talk to attend. We have Giuliano Gianlupi, who is a group member of ours, who will be giving a presentation on how to upload your CompuCell simulations as ready to run NanoHub tools. So if you just want to send somebody to a link that will run your model basically in one click, that would be a good talk to go to if you'd like to start writing some CompuCell simulations and disseminating them as things that anybody can just click and watch and manipulate with the steering panels that James talked about today. And then Priyam will also be giving another talk tomorrow on cell lineage tracing. She'll also talk a little bit more about doing some scientific plotting in that talk as well, if that's something that you've been looking for. And then just to get back to our fourth talk tomorrow will actually be Julio talking about planar cell polarity. He can probably fill you in a little bit more when we turn it back over to him about the abstract for that. So that's all I had. Again, keep an eye out on your email inboxes in the morning for the links that you'll need for these parallel sessions. And we'll make sure not to have the same problem we had today with those. And I think that's all I have. So I will turn things back over to Julio to continue answering some of these questions because I know people have a lot. So thank you all. If you don't have any questions, you're free to go and we will see you
Bharat
tomorrow.
has a question. Bharat has a question.
Yeah, no, I'm just saying for those who don't have questions or who, you know, are not listening in on the questions, you're free to go for today. And we'll see you tomorrow morning.
Yeah, can I ask? Okay, awesome. So I was wondering that I think Darshan pointed it out in the beginning that when you define the width, it goes in all the planes. So if I'm like that, the cell is gonna be a square or a cube. But if I have a 3D model and have my cells on top of something, then I want it to be more spread on X and Y than my Z, because that's how generally in vitro cells behave. Even if I vary my initial conditions, I'm not able to achieve that. As in, if I vary my contact energy with my bottom substrate, it stretches, but my Z kind of starts to chop off, like cells become kind of weird in Z, if that makes sense. Can you tell me a way or some help here which can prevent that from happening?
Okay, so I guess in your particular case, your cells behave in a way that it's not give you a transition to the right initial condition. So probably there's something else going on. But I'll tell you another way that you can write initial conditions. So once you can use the uniform initializer, and you can also use the blob initializer. Okay, if you want to do something very specific, okay, if you basically want to draw the cell at specific coordinate points, there is a way to do that from the Python level. Okay, so you can, let me just share my screen here to show to you very quickly how this is done. And I'm not sure that this had already been covered before, but I'll just use the code that we were working before. Okay, so here on the start function, for example, you want to draw a cell at specific coordinate points. You can use the actually it's better if I share my desktop. Okay. Can you see my desktop now?
Yeah.
Okay, so if you go to, so I'm in the start function because that's where usually when you want to set up your initial conditions.
Right,
So
right.
if you go to CompuCell3D Python, cell attributes, no, cell manipulation, sorry. There is a create cell option and create cell detailed option. Okay, So you can click that, and then you can see that there is an option to actually draw a cell directly on the lattice.
Great.
So you basically need those two comments here, right?
Right.
You first you create a cell object, okay, and then you just set the type of the cell. So it can be of type ECM, for example. So
Yeah.
this creates a cell object, but the cell does not exist in space. Okay. So and then on line 19, you can actually draw the cell. You can make a square of the cell, so you
Right.
can make it at, I don't know. So it will be X, Y, and Z, of course. And what you can do here is you can do at coordinate, I don't know, X from 10 to,
And this will allow me to change the shape to a rectangle or a cuboid, right? It won't
yes,
stay the
yeah,
cube.
because then
Yeah.
on the X can be from 10 to 20, right?
Right. And
But
it
the
can be
Y
different.
can be from, I don't know, um, 60 to 63, right? So
Right.
between,
Got it.
and then very long on the X, right? And if of course this just allowed to draw a parallel. Okay, I do not know the name of the object in English, but a square in 3D that has different lengths. Okay.
Yeah. Yeah. Got it.
Rectangle.
A cuboid.
Rectangle.
Now rectangle is 2D. In 3D, there's another name.
Cuboid. Cuboid.
Yeah, cuboid. Exactly. Okay. In Portuguese, is very different. So
Yeah,
you
I
just
got that.
do this from the cell, but if you don't want to do a cuboid, you want to do more complex, you can just make the skull. Um, okay, it's like in Portuguese, parallelepiped. Uh, you can maybe you want to do
Mm,
like another layer on top that's a little bit thinner. Okay.
Okay.
Yeah. So
I can put this inside a for loop and then maybe make these position multiple of some numbers so that I can have many cells, right? I
yes,
believe
you
that's
can.
how I'm gonna create a layout.
Yes, yes.
Okay, okay.
The other thing
Great.
you can do a loop over uh, visit all lattice
Yeah,
pixels,
yeah.
right?
That
And
makes
then
sense.
you check some condition if is within this and these boundaries, or
Mhm.
is it within this distance from this center of mass here. So you can use that to actually draw a cell that's kind of circular, for example.
Yeah, that makes sense.
And so you can use that. Well, with this you can draw whatever you want. Okay. This comment here does not be a range. It can be, if you just do, for example, 4, 7, and 9, you draw you can write one pixel at a time. Okay. So okay, you got what I want to say,
Yeah,
right?
yeah, I got it. Yeah.
You can
Thanks a
draw by
lot.
hand. Okay.
Yes, yes.
it,
I
it,
can play
it
around
will,
with it. That makes
will
sense.
take some time. Okay. But once you one thing that you can also do, you can use PIF files. Okay. Initial condition Python pixel initial condition files format to
Mhm.
also, once you write your initial condition, you don't have to do this again. You can start simulation by just calling a file that has the all
The values, the
the
lattice
val-
value.
yes, the lattice values.
Okay. Thank you.
Okay,
Thanks, Julio.
no problem.
What I do in the for loop when I draw my cells, basically when you when you loop over all coordinates, you can build any shape you like. If you can translate the shapes into mathematical formulas.
Yeah.
So you can build paraboloids, circles, spheres, ellipsoids, everything using conditions.
Mhm. Yeah, that makes sense. I will implement this. This should work. Thank you.
You're welcome. Yes. Next.
This is more of a logistic question. I've been traveling the last couple of days and been watching the recordings on YouTube, which are like 12 hours delayed. So I missed the hackathon prep session today. Is there anything that I missed that was really important?
So we had a couple more lightning talks, and then we briefly looked at how teaming was going for people.
Okay.
And we were just looking at the sheet to sort of update where the different teams are. So if you've had people join because you presented a project, right?
Yeah, I think I have 5 people that joined.
Okay. Yeah. So if you want to go in, 5 is a pretty good number. Once you get it, 5 including yourself.
5 excluding myself.
Okay. So once you get up to
one of them is like has a really weird time difference, so he's tentative, but
okay. Yeah, once you get up to about 6, that's about as big as a group can be before management gets untenable for a 2-day project. So I would say if you want to go ahead and go into the shared document, just note next to the project title that you're not looking for group members anymore, so people know.
Okay.
That the group is full. Just put group full next to your title. But yeah, we'll have some discussion time tomorrow if we want to talk about project scoping or planning or anything like that.
All right. Sounds good. Thank you.
Yep. So any other questions about anything Julio presented, or course logistics, or hackathon, or scientific philosophy like yesterday, anything? We give people a couple minutes in case they stepped away or are furiously typing a question into the chat and anything like that. I know that's the worst, when you've been like typing a question in the chat for a while, and then the instructor starts to go like, well, if there's no more questions, and you're like, oh. So always feel free, if we're about to move on and you're trying to finish a chat question, always feel free to unmute and just tell us to hold on a second so you can finish typing before we end the meeting.
Just on what I was put in the chat before, I do often get that the cell nucleus just disappears. Is the best way to handle that just to play around with your energy, your different energy terms? Just to try prevent that happening?
Yeah, that would be one way to do it. Do you want to share your screen and show?
No.
What's
Normally, increasing lambda volume is the thing that you need to do.
Okay.
Because if the cell is compressed, is collapsing, that means that the surface energy is large compared to the volume energy. So if you increase lambda volume, that will balance it. If you increase lambda volume too much, the cell will stop moving. But there should be a pretty good sweet spot of a factor of 5 or so over which the cell will move fine and it won't collapse. But why don't you show us, and we could actually do that together? That's a good learning experience, actually. If you're willing to screen share it, we'd be happy to look.
Yeah, sure can. So it's this one here. If I just change, I'll put it back to what it was just to show you. So it's initializing like this, and then the nucleus just disappears pretty quickly. Oh, yep, and easy. Yeah, I do find when adding in a value, I'm not sure if that's actually even relevant anymore. But when adding in a value for this, it also tends to make the nucleus disappear. So yeah, there's just a lot of ones to play around with.
So the smaller the domain, the more likely it is to disappear. And the higher the surface energy of the domain, the more likely it is to disappear. But in general, the one thing that CompuCell should do that it doesn't is give you the typical energies for each of the terms in your plugins. But you need to balance those a bit by hand. But if a domain disappears, then that almost certainly means lambda volume for that domain is too small. And you shouldn't have to push it up by a lot. You could try pushing it up by a factor of 5. If it freezes, then you're too high. And so there should be a region of at least a couple of factors of 2 over which it's motile and it doesn't disappear.
Yeah. Okay, sweet. I'll have a play around with that.
If you are you trying to build a cell like in my videos?
Which videos? Oh, yeah, Pedro. Yeah.
Are you using forces?
I am just starting to implement them, and I'm trying to see if I can manipulate the length to be in the direction that the lamipodia are heading, so that elongates in that direction.
So you're using external potential.
Yes.
Okay, I don't use external potential.
Okay.
It's more interesting, and the cell moves faster as well.
What do you use instead?
I use internal field for lamellipodium.
Okay.
Do you want to see?
Yes, please. That would be amazing.
Okay. So I will first tell you the idea. Try to make you understand what's the idea behind it. So let's say you have 2 cells. Sorry. Yes. Then you have, for example, one you will assign the back type, and the other will be the front type. Okay.
Yeah.
So then you build a field in a way that the field is always 1 in the front cell and 0 otherwise.
Okay.
So you can do this, for example, with extremely high decay outside. Also in the back, extremely high decay everywhere. 0 decay inside, okay? And then you make the cell, or you can, yes, and then you make the cell hold the field at 1, constant value 1, secretion at constant value. And then you apply chemotaxis with a negative lambda parameter towards medium. So this cell will try to copy in those directions. So it is a more distributed force. You don't have, for example, this direction and then everything will try to go there. No, it has, it's a self-polarizing cell. In other words, your polarization is not dictated by a force that you input in the simulation.
Yeah, perfect. That's exactly what I wanted to capture.
So in the videos, for example, actually it's too cringe to show my YouTube, but we always have to go through that song.
Don't be shy, just go ahead.
This one and this one. So all those videos, it's the same mechanism.
Okay.
So this one, there's an external field here. This one as well. So this is the most, look, you have the front, you have the back, and then they move because of the field, because the field is 1 on the front, 0 on the rest.
Okay.
So if you want to implement this, I certainly can help you.
That would be really great. Thank you.
Just wanted to make a quick note. I guess Max already left, but yeah, when you're looking for the videos, the auto-uploaded live streams that go up right after the sessions, you have to click on James's video page. There's a drop-down arrow that says uploads. You have to drop that down to either all videos or past live streams to see the auto-uploaded live streams, because the videos up, the edited versions are only counted as uploads. YouTube hides that drop-down well. So I'll send out that link in the morning too about where to find the auto posted videos.
Probably worth also pointing out that that's not the same channel as the workshop channel, right? That's James's channel.
Right, but that's the
The
James
workshop channel is a stream within my channel.
Oh, I see. Okay, but if you have the link to the workshop,
it's a playlist. The workshop is a playlist within my channel.
I see.
So all of the CompuCell workshops, current and past, are there. All of the seminars related to CompuCell are there. So if you go up to my channel, you'll find all of the CompuCell material. But the link that I've given is for this workshop specific.
And once the workshop and hackathon are over, we will put the edited versions of all the videos together in its own dedicated playlist and send that out to everyone who attended, so that you have easy access to the shorter versions of these moving forward. And by edited, we just mean that we're cutting out the dead air before and after each session. We're not cutting out any content. Any other questions?
And I am trying to figure out a way to make cells move in a random fashion, like the one I showed, but with just one cell, not front and back, like one cell in one field, and then by the way that field distributes internally, you generate the force with just one field and one cell. But I didn't manage to do it yet. That's why I was learning Tellurium, because probably I would have to solve some chemical reactions to produce the polarization in the field internally, but I'm not sure how to approach this problem yet.
I'd be quite interested to work with you on that, Pedro.
So if you have time now, we can. No, I actually I have to keep with the organization team for a while, but we can. I send you a message in Slack after. And then we can meet in Google Meet, for example. And then I show you how to do it.
Awesome. Sounds good.
And I gave links to all of these other social media. I realize everything gets distributed, but we do have this Facebook, LinkedIn, Twitter accounts, which people can use to communicate. the Slack may be more effective, but potentially you could find community people to help with these other ways too.
Just wanted to get you guys' opinion as well. With adding a length constraint to that cell that I just showed you, I would like it to be in the direction that the cell is moving. Is that sort of an inherent property of the length constraint, or is it quite a separate thing?
I would really recommend using compartmental cells if you're going to do that, rather than the length constraint.
Okay, so the one that I showed you, you mean adding like more compartments of the cytoplasm to that?
Right. So because fundamentally, the moment you have a surface energy, things want to be round.
Yeah.
And if you try to stretch them, then you have something called a capillary instability. And that capillary instability means that the cell will start to break up into pieces if you pull on it too much. That's why you can't elongate the cell too much.
Yeah.
In simulations that did that, which we did, for example, with, and there's an option in CompuCell, which is connectivity constraint that you can turn on. There's a 2D fast connectivity constraint. There's a 3D connectivity constraint. So if you want to use the elongation with a large aspect ratio, then you have to turn on the connectivity constraint. That works, but it's slow. It'll slow the simulation down a lot.
I sent you a photo of one with the connectivity constraint on it, and it tends to be like a blob moving, and then it just like it forms like a long string, and then it's just the blob moving. Yeah.
That's the same capillary instability that you're going to get.
Yeah.
And so if you use multiple subdomains, then that's what I think Julio showed. You could string subdomains together to make arbitrarily long objects, and then each subdomain doesn't have such a big difference in curvature between the top and the bottom side, the tops and the edges. And in that case, you don't have the energy problems that you have. And so, stringing things together as a chain of beads, if you wanted to do something like as I say, a long bacterium, filiform bacterium, that's really the best way to do it.
Okay.
And there's some tricks. I mean, AJ was asking, could you make the compartments identical and then make them different? And yes, you could do that. You could have the cells initially, you define 50 compartments. Initially, they have no energy between them, then they mix. And then you gradually turn on phase separation. And that's more or less what Julio did in his kidney simulations. Initially, those cells, apical, basal, and lateral surfaces are identical in properties. Then the cells are basically mesenchymal. There's a hidden set of compartments that aren't doing anything. Then you turn on differences in properties and you gradually polarize the cell. So that's the way.
Okay.
I wish there were a simpler solution, but the problem with geometry is there's so many different kinds of geometry that people want to implement that there isn't a single universal way to do that.
Yeah, that's all good. No, I'll definitely give that a go. Thank you.
Any other questions or comments?
I have a question. Is it possible to define, for example, an energy relative to torque between successive links?
So that's supposed to be implemented in CompuCell. I don't know if it's still there. There's a bending module.
Yeah.
I don't know whether it works.
Okay, I never tried, but it is it seems to be useful if you are trying, for example, to model invagination.
Yes, it's very helpful. So if you want to check that, that would be great. Creating a demo. If you could recreate Andreas Deutsch's old demo of the bacterial movement, that would be ideal. If it doesn't work, we need to know it and fix it.
And Liliana could try, because Liliana is trying to do that that invagination simulation. She could try. I could help her as well.
But there is a bending modulus. It's a little tricky. Defining the bending modulus is actually reasonably complicated, the way to specify it. But I don't know that if it's not there, it's easy to write the plugin to do it. So we should make sure that that's there.